Good afternoon, and thanks for joining us to have a conversation with Will Ho, CEO of IN8bio. IN8bio is a clinical stage biotechnology company developing genetically modified gamma delta T cell therapies for treatment of cancer. The company has 3 clinical candidates and 2 preclinical programs. The company's lead molecule, INB-200, is currently being evaluated in a phase 1 clinical study. And INB-400 is being evaluated in a corporate-sponsored study, both targeting recurrent GBM patients. The company's third molecule, INB-100, is an allogeneic product being evaluated in a phase 1 study as a therapy for leukemia. Recently, the company introduced its exciting platform that can develop off-the-shelf iPSC-derived gamma delta T cells. So to talk about the company's strategy for rest of 23 and beyond, I welcome Will to this fireside chat. Will, thank you for joining us and accepting our invitation to talk to our audience today. So to start off, what are gamma delta T cells and what is their role in the immune system, and why do we think that they can be a therapeutic cell therapy?
Great. Thanks, RK. Thanks for everyone for attending today. It's really nice to be here. So look, at IN8bio, we are focused on developing gamma delta T cells. We believe that the gamma delta T cell is a unique subset of immune cell or lymphocyte that can really have an impact in targeting cancer cells. If you think about it, the gamma delta T cell is a cell that bridges between the innate or the early immune response and the adaptive and the memory immune response. And as such, it has properties across both. If you actually think about it, before we had specialization of the different white blood cell components, the gamma delta T cell was one of those early, older immune cells that can somewhat do it all. But today, when our team thinks about the advantages of the gamma delta T cell, it's twofold. One is that while it can kill directly, it also acts somewhat as the conductor of the immune system. It can interact with all the different parts of the immune system, whether they are the natural killer cells or the NK cells, part of the innate immune system, the alpha beta T cells, the B cells, the dendritic cells, the macrophages, and other. And so it can elicit all different parts of the immune system to come and attack the tumor. Importantly, one of its other features is that it has the natural ability to distinguish between tumor tissue and healthy tissue. And we think that's important as we move onwards and beyond the current generations of CD19 and BCMA CAR Ts. Those cell therapies, I can drive aplasia or wipe out the entire B cell compartment or tissue compartment. As we get to the targets for solid tumors and the more complex leukemia targets, we can't do that. You need all the different components of your liver, your lung, your brain, and other parts of your body. And so we really do need to be able to distinguish to make sure we have a safe therapeutic window.
Very good. So how do you differentiate gamma delta T cells from NK cells, TCRs, and CARs? And can we also talk about the advantages that they have in terms of efficiency and safety?
I just touched on it a little bit, but let's go back to 2013 when Kite and Juno came public, right? The first CAR Ts developed at the University of Pennsylvania and Memorial Sloan Kettering really focused on the adaptive or the memory immune system. And there we're using the alpha beta T cells. Quite frankly, one of the reasons why we use the alpha beta T cells was that they're plentiful. If I take your blood, 70% of the white blood cells in that blood are alpha beta T cells. And so I can get a lot of them fairly easily. On the other hand, the gamma delta T cells encompass only about 5% of the total white blood cell population. And so it's more complex to get the cells to expand them to sufficient numbers to get a dose. But ultimately, as I mentioned earlier, it's that ability of the gamma delta T cell to interact with all the different components of our immune systems to ultimately drive the response. When we think about it, think about a symphony and concerto. When you go to the symphony, it's not a single instrument that creates the music. quite frankly, if I only played the percussion instruments, it'd be quite boring and maybe sometimes quite awful. But it's how the instruments come over time and where I get the brass, the strings, the percussion, and how they interact each other and their timing relative to each other that ultimately creates the music. Along the same way, our immune system functions by driving different cells at different times to ultimately attack and eradicate the cancer cells.
Very good. In terms of gamma delta T cells, there has been a debate going on, regarding persistence and expansion of these cells. There are thoughts on either side of this argument. In terms of your thinking and IN8bio's thinking, where do you guys stand?
Yeah, so I'm really excited about gamma delta T cells. As I'm not going to beat around the bush, as we know, cell therapy has been a very difficult environment over the last 8 to 18 months. In particular, allogeneic cell therapies has been difficult as people try to get towards off-the-shelf therapies. Many in the audience and even in this room may know that for a long time, I was actually on the buy side. I was an investor. I was actually an investor in NK cells early on. But one of the limitations of the NK cells that I realized was their lack of persistence. Unfortunately, as the clinical data has come out over the last 6 to 12 months, that lack of persistence has translated into a lack of durable responses. As I said earlier, the gamma delta T cells bridge between the innate and the early immune response and the adaptive and memory immune response. And so they do have persistence. In fact, last year, Carl June at the University of Pennsylvania published a paper in Nature that demonstrated of the 2 patients that they first treated with their CD19 CARs, one of those patients, 10 years onwards, 50% of the CARs that are still expressed in their body were actually gamma delta T cells. In our own allogeneic study, what we call INB-100, we presented at the European Society of Bone and Marrow Transplantation, or the EBMT conference in April, for the first time, a donor-derived infusion of cells. We showed that we still found gamma delta T cells out at 180 days, and that the levels of gamma delta T cells increased from 60 to 180 days. So we're actually demonstrating not only persistence, but in vivo expansion of these cells.
Okay. So to talk through your clinical programs, I'm starting off with INB-200, which is the lead molecule of yours, currently being evaluated in an investigator-sponsored phase 1 program for treatment of GBM. And you have released some data from this from this study. So what can you highlight some of the data? What has been released so far, and what's the next set of data that we should expect?
Yeah, great. So INB-200 is our autologous, meaning it comes from the patient themselves, genetically modified gamma delta T cell program for solid tumors. As you're aware, not too many people have actually designed cell therapies for solid tumors and have been very successful to date. This particular program is in the upfront setting, so newly diagnosed GBM patients, and it's in combination with chemotherapy. We use the conventional chemotherapy to prep the tumor microenvironment, but also to force upregulate a signal on an immune signal on the surface of the glioblastoma cells that allow the gamma delta T cells to identify residual GBM cells, even if they're resistant to chemo or they're part of that so-called cancer stem cell compartment. We actually presented some data at ASCO. At ASCO, we actually had an oral presentation. We were one of only 4 companies out of hundreds in the CNS tumor section to get an oral presentation. It's a phase 1 study. At that point, we had demonstrated that we are currently in the 3rd cohort of patients. And I believe we had treated at that point about 8 patients. The vast majority of our patients that were treated had gone beyond their expected median progression-free survival. Quite a number of them had gone on and went beyond their expected overall survival. Unfortunately, early in the trial, we had at least 3 patients who passed away for unrelated causes to GBM. Two of them were anticoagulation issues with pulmonary embolism and myocardial infarction. Part of that may be because we were enrolling patients during the early parts of COVID as we look back, but we think we're doing quite well. We expect to have an update later this year, probably sometime around November 16th to 19th at the Society of Neuro-Oncology. And we'll give an updates into the clinical data and the patients that we've enrolled thus far. What we have said this year is we do expect a complete enrollment in this particular trial. And I think ultimately what would be positive is if those patients who we treated continue to go and survive, the longer that they can go progression-free and the longer that they can go with respect to their overall survival, ultimately the better it is. But importantly, we know when we treated patients with a single dose, eventually all of those patients relapsed, and 2 of them ultimately died of progression of disease. As we go from 1 dose to 3 doses to 6 doses, what we ultimately expect is to see longer and longer time to progression and longer overall survival. We know in many solid tumors, and in particular glioblastoma, overall survival is ultimately related to the time to preventing tumor regrowth and the depth of response. I think our unique therapy allows us to get deeper, targeting those compartments that are resistant and aren't identified by traditional care or aren't resectable by surgical resection. And the continued repeated dose allows us to continue to keep the tumor regrowth in check.
I know you answered this question to a certain extent, but want to see if you have any additional commentary, because GBM is been a beast in terms of developing therapies against it. So I know you gave some reasons why you think INB-200 or gamma delta T cell therapy is the right way to do it. Is there anything else that you think makes you more comfortable and confident that this is the way to go?
So look, I wouldn't say just GBM is a beast. In fact, I would say all of cancer is a beast. Cancer is a horrible, disease that impacts all of us and all of our lives. As we looked, I know a lot of people look at GBM and says, oh, that's too hard or that's difficult. One of the problems, though, of GBM is that most companies and most people try to dose peripherally. So they dose through intravenous or orally into the gut, assuming that their therapy is going to cross the blood-brain barrier and your therapy is going to be active there. As I said earlier, I was a long time buy-sider and sell-sider. And one of the things that I learned over my career is as I made mistakes, if you get the PK and PD wrong and your therapy isn't where you think it is, you're not going to have any effect. Our immune cells are like our soldiers. If our soldiers aren't on the battlefield, they're not going to do anything. And so one of the keys for me, when I look at trials and look at programs, is how do I reduce the number of variables that are going to be the landmines that I step on that causes our treatment to fail? In particular, around cell therapy, T cell trafficking is a big issue, right? We have to get the maximum number of our soldiers onto that battlefield. When we infuse intravenously, many of those cells get stuck in the lungs. If they're in the lungs, they're not in the brain. By going to the brain, the patients, 95% of them get a surgical resection. We can put a catheter that bypasses the blood-brain barrier and goes directly into the tumor bed. And that way we know when we infuse our cells, we're putting them on the battlefield such that we can generate as high of what we call an E to T or effector to target ratio. That's the ratio of the number of soldiers that I have versus the number of tumor cells. The higher that gets, the more chance I'm going to have to ultimately killing that tumor cells and eradicating it.
So moving from 200 to 400 and 410, these are the 2 assets that you have, which are allogeneic and autologous therapies against GBM that the company is developing. So what are these 2 programs? Why 2 programs? And what's the clinical development strategy here?
Great. So, when we started it, when we launched the INB-200 program, we actually launched it as investigator-sponsored studies, or ISTs. Generally, a lot of companies did that 20 years ago. They're faster, they're cheaper. The challenge is making sure that you have a robust database, which we have. We directly negotiated to be able to get access to those databases. INB-400 is actually our first corporate-sponsored IND. We often had a lot of criticism of people who said, well, that's an IST, you didn't do that, or you can't get through the process for a corporate-sponsored IND. So last fall, we filed our first corporate-sponsored IND. We actually cleared FDA IND clearance in 30 days on first pass. We cleared Pharm Tox, CMC, and clinical regulatory review on the first pass. And so this is a very similar design structure to the INB-200. We'll be repeating that, but it'll be multi-center. It'll be national in nature. It'll have 15 centers around the country. On our website, you can see some of the centers. The first one will be University of Louisville. We'll have UAB again, Duke, UCSF, Cleveland Clinic, a number of other centers, Moffitt, and a number of other centers all around the country. Those programs are to confirm the signal that we believe we're observing in the phase 1 program. And that program is actually going to be in 2 parts. The first part, in which we have said we're going to initiate enrollment in 2023, is the phase 2 portion of an autologous program, meaning the cells are derived from the patient themselves. The holy grail in cell therapy is ultimately to get to an allogeneic and potentially off-the-shelf therapy for solid tumors. And so the second portion of this 400-410 program is generating an allogeneic or donor-derived cell therapy for the solid tumor. The advantage of the donor-derived cells is you can take cells from a younger individual who doesn't have cancers, so they should ultimately have greater potency, greater killing ability, and be more robust. But having a donor-derived cell has risks, in particular the risks of graft versus host disease, or GVHD, or even the risk that the host's own immune system will kill or eliminate your cells in what's known as host versus graft. And so that program is split in 2. As of the end of last year, the FDA gave guidance. Similar protocols between allogeneic or autologous programs now require a separate IND. We are seeking to file an additional IND for the allogeneic program. And ultimately, this program will test in glioblastoma the potential safety and efficacy of autologous and allogeneic in both the frontline and the relapse refractory setting. A very comprehensive program. In fact, we think there's a lot of interest in the agency because most people, or I don't think anybody, has tested that. As to what the specific indication and whether donor-derived or patient-derived cells are ultimately better.
So on the third program is the INB-100, which you're testing in leukemic patients. So what's the features of INB-100? And you also have published some data on this molecule, INB-100. So what can you highlight some of the data, and also what's the next data set that we should be seeing?
Yeah. So INB-100 is actually the first allogeneic or donor-derived gamma delta T cell program that the FDA had seen. We're the first to give a donor-derived gamma delta T cell into a patient. The FDA was actually quite concerned as to the relative risk of graft versus host disease. These are actually going into patients, the highest risk group, who are undergoing transplantation. They're of high risk because you're ablating their bone marrow and creating a large niche while infusing the cells. And in this particular case, we had enrolled patients with leukemia undergoing haploidentical stem cell transplantation. About 5 to 6 days after neutrophil engraftment, they get an infusion of gamma delta T cells. At the BEBMT conference in April, we presented our first data. At that point, we indicated 100% of our patients remain in remission. Complex cytogenetic AML patients out as long as 3 years in remission. These patients generally relapse at about 50%. In fact, with the complex and high-risk cytogenetics, that relapse rate can be as high as 80 to 90%. And we had, as we had announced earlier, we had dosed, I believe it was about 7 patients at that point. All of them remained in remission. We have given guidance. We will complete enrollment this year.
Yep.
We expect to provide an update at the ASH conference
Mhm.
as to the patients and the durability of remissions and the length of those remissions.
So the last program that I want you to discuss with us is the non-signaling CAR T molecule, which is the INB-300 program. So why a non-signaling CAR T? And what are your thoughts around this? And I know you have a development strategy set up as well. So what's that?
Yeah. So look, the challenge with CAR Ts is they allow us to have the immune cells stick to the target better. But the challenge has been you don't discriminate between your healthy tissue and your tumor cells. Outside of the current 2 CAR Ts with CD19 and BCMA, many of the targets such as CD33, CD123, and the solid tumor targets are challenging because they're expressed on other cells. In particular, you may have seen the news yesterday, one of our competitors announced a layoff. Unfortunately, they laid off about 176 of their employees, partially because they're on clinical hold with the CD33 targeting CAR T. Unfortunately, not only is that target expressed on leukemic blasts, but it's also widely expressed on CD34 cells, or what are known as your hematopoietic stem cells. You need your bone marrow. If I wipe out your bone marrow, you're going to unfortunately kill the patient. And so the question is, how do you create a wide therapeutic index? Some people are trying to genetically engineer the bone marrow so they don't express that target, transplant that patient so that you can use the CAR. But we believe that's risky, because patients die of their transplants. And so if we can create a CAR T that actually has its own natural ability to distinguish between healthy cells or healthy bone marrow and leukemic blasts, that becomes safer and potentially of greater efficacy. And so we believe the gamma delta T cell can do that. Our INB-300 program is a non-signaling CAR that is able to discriminate between healthy and tumor tissue. Against the CD19 target, we showed data at AACR. We had 90% plus killing against the leukemic cell lines and only 5 and indiscriminate against non-CAR expressing cells on the healthy B cells. First that I know anybody has demonstrated such a wide therapeutic index. And so we have a broad platform around gamma delta T cells. We're excited about all the progress we've made thus far. And we think this fall is going to be an exciting time as we present additional data at medical meetings. We're targeting SITC in early November, SNO in mid-November, and wrap up the year at ASH. And so thank you, everybody. And look, we're excited about what we have.
Thank you. Thanks, Will.